5 minutes for discussion. Just in order that the speaker is not disturbed, but since we have time, then 3 minutes are there. We will, the speaker will show something like this, that 3 minutes are left, so you know that you have to come to a conclusion. So, welcome friends for this extremely academically sound meeting. And I have the privilege of inviting Professor Iain Mathieson from University of Pennsylvania, and he talks about special cases and general principles of natural selection in humans. So, Dr. Mathieson, please.
Thank you. Well, thank you very much to the organisers for the invitation and for the honour of delivering the first talk this morning. My name is I'm Professor of Genetics at the University of Pennsylvania, and my lab works mostly on looking at human population genetics, particularly looking at patterns of natural selection. That's what I'm going to talk about today. So as people are mostly familiar, humans have spread out through a vast range of environments since our origin in Africa maybe 100,000 years ago. And you know, those differences in environments include things like the development of agriculture, the development of different social structures, and really, if you think about the environments in which humans, anatomically modern humans would have evolved, perhaps in the African savannah, it's really extremely different from the environments that we live in today. This is the city of Philadelphia. Okay. Now, a lot of the sort of demographic history of this expansion has been elucidated in the last few years by using ancient DNA. So ancient DNA has really taken off, I would say, in the last decade from its origins, maybe in 2010. The number of samples has really increased dramatically, largely due to the development of new technologies. As you can see, this is a map showing the location of these 10,000 samples. As you can see, it's mostly Eurasia, although we're starting to generate data from other parts of the world. Now, if we're thinking about detecting natural selection, thinking about looking at allele frequencies. So there are really 3 things that can cause allele frequencies to change in a population: natural selection, which is the thing we're looking for, genetic drift, and admixture, so when 2 distinct populations mix. And those are really confounding factors that we're trying to ignore. Now, there are really 3 ways of looking for the effects of natural selection in the population. One is by looking at differences in haplotype structure across populations. We can do that with modern data as well as ancient data. One is by looking at highly differentiated allele frequencies. I think this figure will be familiar to some people in this room. And one of the things we can do with ancient DNA is actually not to take these indirect approaches, but to just measure allele frequencies directly as a function of time and see whether they change faster than we would expect due to genetic drift. Okay, so that's using time series. And the first thing I'm going to talk about is how we can use that approach to look at natural selection in a particular context where we don't have to worry about admixture. So really here we're just trying to separate the effects of selection and genetic drift. Okay, so the first project I'm going to talk about is a paper that was published a couple of years ago. This is a collaboration with Jonathan Terhorst at the University of Michigan. So we're looking at natural selection in Bronze Age Britain. Why are we looking at Britain? It's not necessarily because Britain is the most interesting place in the world. I think it is, but that's not why we're looking at it. It's really just because we have a lot of data. So here's a figure showing the histogram of the number of ancient humans with genome-wide data from Britain in the last 4,500 years, and you can see we have a pretty good sampling. And then, of course, we have a lot of present-day samples. So this is where they're from. The other reason we look at Britain is because at this time period is because there hasn't been much change. Okay, so here is now a PCA plot. Each grey point is a present-day person from Europe, and you can see sort of the structure of Europe, Northern Europe, Southern Europe, and neighbouring regions here. And the point is that if you look at present-day British people in light blue, they all cluster very closely, and they also cluster very closely with the ancient Britons from this time series. Okay, so in the last 4,000 years, the population of Britain has been relatively constant. There's been little migration from outside. These are a bit more spread out because they're ancient samples, so they have more coverage. So we can, in this case, we can ignore the effects of admixture and really just focus on the effects of genetic drift. Okay, so what we do simply is to measure the allele frequencies over time, and then test whether at each SNP in the genome whether that SNP has changed in frequency faster than we would expect given genetic drift. So here's the results of that. So here each point is a SNP ordered along the genome. Each chromosome is colored in a different color. The y-axis is the negative log p-values. When you see these kind of low p-values here in most of the genome, that means that those SNPs are behaving as we would expect given genetic drift. Okay, so they're just behaving neutrally. And when you see a big spike here, that means that there's a lot of SNPs in those regions which are changing in frequency faster than we would expect genetic background. Okay? So that is the sign of natural selection. Now, I've labeled these with the gene names that these SNPs are sort of largely either regulating or encoding regions. And actually, it turns out we know what all of these genes do, so these will be familiar to many people. SNPs associated with lactase persistence, the ability to digest milk as an adult, SNPs associated with skin pigmentation, vitamin D metabolism, and then a lot of SNPs in this region, the HLA, which has a lot of immune function, but particularly our lead SNPs are largely associated with things like celiac disease. Okay, now, looking at these, you might think there's a sort of variety of different things going on, but actually, it turns out there's a parsimonious explanation for all this selection, and that is that the weather in Britain is really bad. So I'm from London, I can tell you this is accurate, this is from a newspaper earlier this month. There's really very little sunshine. So when people migrated to Britain from southern Europe or from Anatolia, from the Near East, where they've developed farming, they found themselves in a region with very little UV radiation, leaving them most likely vitamin D deficient. Now, one of the consequences of that is the inability to absorb sufficient calcium, leading to bone deformities, obviously very strong selective pressure against that, particularly pelvic deformities. Okay? So in fact, a kind of parsimonious explanation for all this selection is that really, by far the strongest selective pressure in this time period in Britain was due to vitamin D and calcium metabolism. And this was actually the original hypothesis proposed for selection on lactase persistence. Other people have ideas about what that might be, but I think our data would support that that really is the strongest selective pressure. Lactase persistence allows you to consume milk, which contains both calcium and vitamin D. Lighter skin pigmentation allows you to synthesize more vitamin D. And actually, even today, celiac disease is a risk factor for osteoporosis because it prevents calcium absorption in the gut. So again, in this case, this actually looks like a relatively simple selective pressure. Actually, we can be a little bit more sort of general. So if we look at other complex traits, here at each point is a complex trait from UK Biobank. So this includes things like weight and other metabolic traits. And the x-axis is showing you the correlation between the effect size for each SNP in the GWAS and the selection coefficient. Y-axis is the p-values. It's kind of like a volcano plot. And you can see that really the only thing that's significant is skin pigmentation. And actually the largest effect, although it's not significant because there aren't that many SNPs, the largest absolute effect size is calcium metabolism. Okay, so again, it's consistent with the fact that those are really the strongest selective pressures and everything else is not really affected. Okay, so that's Britain, relatively recent history, but obviously that's just one example. What about the rest of the world? Okay, so this is work we have in preparation by Laura Colburn, who's a postdoc in my lab, as well as Jonathan. Okay, so what I didn't tell you is that there's been lots of work on natural selection using ancient DNA in Britain. So everyone publishes lots of selection scans. You might sort of disagree about some of these specifics, but generally, the more things, the more samples you look at, the bigger the sample size, the more things you find. Okay, so we have a pretty good understanding of what's going on in Europe, but not so much in the rest of the world. So what we did is we went and collected all the ancient DNA that's been published from different parts of the world, and we kind of arranged it into these broad groups. Okay, so you can see obviously we have the largest sample in Europe, but we have relatively good sample sizes in East Asia, for example, in the Americas and Africa, and a little bit in South Asia, although you'll notice that, so here the present day samples are in crosses, so these are from the 1000 Genomes Project, the ancient samples at these points, and you can see that the data are really only from present day Pakistan and North India, so relatively limited data so far. Okay, now, in the first part of the talk, we ignored the effect of admixture. But when we're looking more globally, and here we're looking sort of on a timescale of about 10,000 years, we can't ignore that. Okay? So, for example, we know in Europe that in the last 10,000 years, there have been two major migrations, one from Anatolia and one from the Pontic-Caspian steppe here. which basically largely make up the ancestry of present-day Europe, to the point where both present-day and ancient populations can be modeled as a mixture of these three distinct ancestries. Okay, so we can't ignore the effect of admixture, because admixture is really dominating allele frequency differences in these populations. And that turns out to be true basically in the rest of the world. So for example, here's what that looks like in East Asia. So here, again, each cross is a present-day population, each point is a site with ancient data. And you can see, for example, we're modeling those individuals as mixtures of unobserved populations. So each line here is a single individual. And you can see, for example, that these ancient Japanese populations all have this sort of dark green ancestry. And then if you look at present-day populations, you know, present-day Japanese people have some of that ancestry, but actually most of their ancestry is this kind of light green color, which corresponds to northern East Asia, and that's again due to another agricultural expansion associated with spread of rice farming. Okay, so we can break, you know, and this turned out to be a sort of general pattern in different parts. Okay, so here's a sort of schematic of how the approach works. So we model both the ancient and present-day populations as mixtures of unobserved source populations. So we don't have data from these populations, but we do have data from some ancient and present-day populations. And we estimate these genome-wide mixture proportions. So that's sort of our null model. And then what we do is we look at each SNP in turn, and we simply ask, are the allele frequency proportions of that SNP consistent with these genome-wide? Okay? And I'm not going to go through the details of the statistics of that, but we can write down a likelihood and we can get a p-value. And then we actually sort of post hoc correct that p-value for the effects of genetic drift. So in the first case, we were just looking at genetic drift as a confounding factor and ignoring admixture. Here, we're looking at admixture as the main confounding factor and then sort of also trying to correct for genetic drift. I think admixture is more important. Okay, so what does that look like in practice? So here's an example. This is the FAD1 locus. This is a gene involved in fatty acid metabolism. And here's what the demography looks like. So these are the ancient populations. These are the present-day populations. They're all mixtures of these unobserved source populations. This is where they are on the map. And if we look at the allele frequencies, so here, this is showing the allele frequency of the most strongly selected allele. The black points are the observed frequencies. And the red stars are what the observed frequencies would have to be in order to match the genome-wide admixture proportions. Okay, so what you can see is that this is telling you in the ancient Northeast Asia, in order to match the genome-wide proportions, the allele frequency of this particular SNP would have to be higher than it actually is. Okay, so you need to sort of flip that round in your mind. What that means is that that has been positively selected over this time period. And we can also look at the time series directly. So here we're sort of modeling allele frequencies separately in each of these source populations. And you can look at the allele frequencies over time. So here, I'm afraid, time is going the wrong way. This is the present, this is the past. The allele frequencies in these different populations, but you can sort of see that actually in the northern component, which is here, it's like the allele frequency is increasing really fast. So again, this is sort of consistent with the fact that it's really selection in northern East Asia that's driving this effect. We can also estimate the selection coefficients. We get the selection coefficient seems to be increasing over time. Okay, so we run this on every SNP in the genome, or something like 1 million SNPs in the genome across all these populations, and we find, you know, different numbers of significant hits in different populations. Now, these differences are largely just due to differences in statistical power, due to sample size and the complexity of demography. So, for example, we find 9 loci in Europe, 4 in East Asia, 3 in the Americas, 1 in South Asia, and actually we don't find anything in Africa. Not because there's no selection in Africa, it's just because the demography is more complicated, we have a small sample size, and we don't really have much power. So one observation is, though, is that actually none of these things we find, or very few of them, are actually shared across populations. So most of the really strong signals of selection are actually independent in different parts of the world. And even if you sort of take these top hits and then you look in the other populations, see if they replicate, there's no significant signal. So really, all of these signals appear to be largely independent, with 2 exceptions, and that is actually shared signals between Europe and South Asia. Okay, so we see 2 loci which are both shared between South Asia and Europe, one at lactase persistence and one, this is actually a similar sounding but different gene, also involved in skin pigmentation, light skin pigmentation. And actually, I was very happy to be able to come here and talk about this because, in fact, we already know that these are shared between these populations, thanks to two papers about 10 years ago. These are actually two of my favorite papers that were very influential on me when I was writing my PhD. And actually, both of the lead authors of these papers are speaking this morning. Many of them involved are here in this room. Okay, so, in fact, we already know that these two loci are shared. It's nice to see that confirmed by the ancient DNA. What the ancient DNA does give us that we didn't necessarily have before is a sense of the difference in timing. So here what we're doing is, so we're sort of modeling South Asian populations as a mixture of sort of ancient North Indian and South Indian-like populations. So this is a distinction sort of discovered by Dr. Thangaraj and others. But basically, what you're seeing is that for lactase persistence, the allele frequency is increasing in the people with ANI-like ancestry, so really largely in North India. And again, that's consistent with the present-day populations. If we look at SLC45A2, actually the light pigmentation allele, it's interesting, it's actually not really changing in frequency within the ancestry backgrounds, but what's happening is that the actual ancestry proportions are shifting. Okay, so the ancestry proportions of this locus are deviating, and we actually see the same thing happening in Europe at this time. That's actually something we know, again, also from this 2019 paper, again by Irani and others. So basically the point is that, you know, in terms of looking at natural selection in South Asia, ancient DNA is not really at a point where it's teaching us things that we didn't already learn from looking at present-day populations, largely because limitation in the sample size and the location of samples. I know people in this room are working on increasing that sampling, and that's really going to drive things forward to the point where we can discover novel things. Okay. In the last minute or two, in the very last part of this talk, I'm going to talk a little bit about polygenic selection. So what I've been talking about so far is the case where we're looking for really like single variants which are strongly selected. So like if you have the lactase persistence allele, you have lactase persistence. If you don't, monogenic. Okay, it's a very large effect. But actually most human traits are not like that. So what we want to do is say whether we can look at selection on complex traits. So things like height, diabetes, diseases, and all of those things. And again, I'm not going to go too much into the statistics here, but basically what we do is we just do exactly the same analysis. Instead of looking at the allele frequencies of individual SNPs, we look at combined allele frequencies of all of the SNPs associated with the trait in genome-wide association studies. Okay. So we sort of just count up the frequencies, what proportion of the time do you have each individual carrying the trait increasing allele, and then we use that, treat that as an allele frequency in our calculation. Okay, so we've done this for a number of traits. I'm just showing you sort of the most interesting here. So here, each point is a trait listed here. Now, if you look to the right of this figure, points on the right here, and this would be a very significant p-value of 10 to the negative 8, those are traits where the allele frequencies are not consistent with the genome-wide mixture proportion. So exactly as we were detecting selection before, for example, SNPs associated with rheumatoid arthritis are not consistent with the genome-wide admixture proportions, and we take that as a signal of directional selection. Okay, so in one or more of these global populations, there's been selection pushing rheumatoid arthritis risk. We don't actually think the selection on arthritis, we think that's probably selection on other immune-related things, probably pathogen resistance, but that's pushing autoimmune diseases as well. We see skin pigmentation, we see tannin response, and asthma is actually autoimmune, similar to rheumatoid arthritis. Now, in the other direction, we see traits where allele frequencies are too close to the genome-wide admixture proportions. Okay, so that's sort of hard to think about because it doesn't really, that can't really happen with a single locus. But basically, what this is saying is that the genome frequencies, the allele frequencies of the trait-associated allele are too close to the genome-wide proportions than we would expect through random chance, suggesting that there's some force keeping them at the same proportion. We call that stabilizing selection. So this is selection trying to maintain trait at a particular level. So actually the classic example here is birth weight, which as you can see is shifted in that direction. And that's actually because of maternal fetal conflict. So like the baby wants to be as big as possible, but like the mother doesn't want it to be too big. So there's this kind of conflicting selective pressure. But actually, we see similar things on diabetes, and actually also really strongly on waist-hip ratio. That's actually entirely driven by female waist-hip ratio. Okay, so we can have a look at this. We look at it, and now I'm splitting this up by population, but for example, we see, again, skin pigmentation, tanning response, and autoimmune traits in most populations. You shouldn't really interpret the difference between these points as meaningful, because again, that can also just reflect a difference in power, showing you that they're all deviated in the direction. Similarly, if we look at waist-hip ratio, again, you can see waist-hip ratio is sort of like significant in most populations, specifically female waist-hip ratio, but actually male waist-hip ratio is like right in the middle here, has no selection at all, it's just behaving neutrally. So again, this is likely something related to pregnancy and childbirth. What it is, more generally, we don't know, this is sort of an open question, but again, this seems to be one of the more general principles of selection in humans. Okay, so that's sort of the talk. What we see in different populations is largely we see very strong selection in pigmentation, the immune system, some aspects of diet, but largely skin pigmentation and immunity. We see very strong selection on stabilizing selection on female waist-hip ratio, plus a few other traits. But one of the takeaways that we've got from doing this analysis is that really the ancient DNA selection scans are still very underpowered in many parts of the world. It's largely limited by sampling. In Europe, I think that's probably not the case. I think in Europe it's limited just because there's so much, now the sample size is big enough that we can actually find basically all the selection, but that's not true in other parts of the world. Okay, I'd like to thank my co-authors on the papers I presented today, my lab at Penn, and of course funding from the NIH. And again, I'd like to thank the organizers of this meeting for inviting me and for hosting such a wonderful meeting already. Thank you.
Thank you very much for such a illustrative talk, and I'm sure we can make questions. Please.
So when we say natural selection, when we see what the definition of natural selection is, the survival of the fittest. That means whatever traits or what we call the diseases confer some kind of protection to the current environment. That's the reason why there is these selection. That means diabetes is more on right, means maybe that is correlated with whatever lifestyle or environment that we are having. Accordingly, same way you're saying that rheumatoid arthritis is slowly going that way. That means they're being picked out. That means it may occur, right?
I think really probably what that represents is like some kind of evolutionary mismatch. So, in the past, perhaps there was some disease and there was selection against that disease, and now that disease perhaps is no longer so bad. And exactly. And so now what you're left with is just this maybe like overactive part of the immune system, which is kind of bad if you don't have the disease. And we see that in lots of other cases as well, like with malaria resistance, things like APOL1 and kidney disease, like, very common, like the things that are sort of strongly selected and apparently kind of negative are because of selection against disease. Exactly. So, the change in environment is very, very dramatic.
And you started your talk saying that the lactose tolerance of the Britons might had something to do with the environment, old environment. But if you see geographically, Japan also is almost on the same line. However, there's a lot of lactose intolerance. So how do you say?
Yeah, so I think that always sort of puzzled me again ever since I read Irani's paper in grad school. I think now what's becoming clear is that in a lot of parts of the world, people have adapted through cultural adaptation. So people consume a lot of fermented milk products, and then they don't have the issue. So I think it's sort of there's more than just genetic adaptation, and I think particularly with a lot of these dietary related things, that's what's driven the change. But I think it's still kind of a good question. It's still sort of an open question.
Yeah, I should take the mic. Yeah, please. Okay, in between and let me ask.
This is really a
good idea.
Definitely. There is no question. I'm just giving some comments, right? So I'm Arup from Calcutta University, Department of Anthropology, because it's a very interesting topic that you are talking about, because all population genetics and some anthropometry present. So you talked about anthropometry, right? So anthropometry having a kind of wide point of understanding in the non-genetic factors. Because if you have the socio-demographic conditions, it's better because of the birth weight, So there is prenatal care of the mothers is very much important and affected in the birth weight. So if you go for twin studies, this is the heritability is very much high in case of the stature in the genetics. And about the WHO, you are talking about the hat patterning, and which has really a sexual dimorphism. So females are having a fat pertaining, they used to have the more fats around the hips.
I think we can give suggestions also.
Yeah. So just is the point, and what the question is this: you have taken the drift and admixture in a way that the drift is